Welcome to the next lecture of our course on environmental biotechnology, and in this particular lecture we are going to discuss about the microbial interactions with heavy metals and metalloids, and this will of course include the radionuclides also, and we will learn how bioremediation of these toxic compounds or toxic metals can be done through the microbial action. So, now the basic concepts those will be covered in this particular lecture will be the bioremediation of metals. So, we will get ourselves introduced to the basic concepts that will highlight the importance of the microbial biocatalysis in or catalytic mechanisms in bioremediation of metals. A basic concept of metal microbe interaction will be discussed, and finally, the metal bioremediation ways to achieve the remedial goals, and some of the aspects of metal microbe interaction also will be discussed in detail. So, bioremediation technology, as we have already discussed, uses the microorganisms to reduce, eliminate, contain and transform contaminants in soil, sediment, water or air to benign products. So, this is a broad technology, and as we already have learned that it includes not necessarily only the kind of detoxification or disappearance of the molecule or the complete removal of the molecule or the contaminant, but also it includes the reduction, elimination, containment and transformation of the contaminants. And in the last 3 decades, there have been an increase in the types of contaminants to which the bioremediation is being applied. Almost all kinds of environmental pollutants are being subjected to bioremediation, particularly using the microbial systems, so called microbial bioremediation. And these include the solvents, different explosives, polycyclic aromatic hydrocarbons, polychlorinated biphenyls and metals and radionuclides. So, with respect to metal and radionuclides, we are going to have this lecture, because the other organic solvent discussion we have already concluded. Now, bioremediation works by either transforming or degrading the contaminants to non-hazardous or less hazardous chemical or less hazardous compounds that we already know. Now, when in a contaminated environment, we have the microbial communities, microbial cells which are capable of acting on the pollutant molecules. The pollutant molecules like A is expected to be converted into the compound B, or maybe A will be converted to compound C. Now, there are actually 2 distinct types of biological transformation reactions catalyzed by the microorganisms, which are naturally present in the contaminated environment. And by virtue of the natural catabolic abilities which are basically due to the enzymes and their intrinsic potential to deal with the toxic pollutants, the pollutants might undergo the biotransformation reaction, or they might undergo the biodegradation type of reactions. During the biotransformation type of reactions is kind of an alteration of the molecule or atomic structure of a compound occurs. So, during biotransformation reaction, the compound may not undergo any kind of breakdown or the oxidation in terms of the complete mineralization kind of things, which we perhaps see in case of an organic pollutant biodegradation, but the particular pollutant molecule like pollutant A may be converted to some form which is possibly having less toxicity or having an altered chemical structure, chemical behavior, and that could be useful for environmental control, environmental remediation purposes. So, these are the 2 different types of biological transformation, rather I will say, which are expected from the microorganisms. One is basically the degradation of the compound, which is more often is seen in case of the organic pollutants, where the organic substances are broken down during the course of the microbial interaction. And as we have learned earlier that these are basically manifested by the oxidative metabolism in most cases. In some of the cases like with chlorinated solvent etcetera, reductive dechlorination or reductive dechlorination, dehalogenation type of reactions might be also there. So, essentially these are biodegradation type of reactions, but everything is not subjected or cannot be biodegraded, because for example, the heavy metals cannot be degraded, but the heavy metals and similarly different other pollutant molecules can be converted into in terms of their chemical structure, atomic structure, so that their environmental fate, environmental toxicity, their mobility etcetera are altered. So, often in microbial bioremediation process, we need to rely on such kind of processes as well. So, these are the two basic types of processes which are basically deployed by microorganisms, and these are found to be more useful for developing the bioremediation processes. Now, bioremediation of metals and metalloids and radionuclides relies on different yet versatile micro-metal interaction, because one of the fundamental reasons that the microbes they need to deploy a separate and very specialized set of reactions in terms of dealing with the heavy metals or radionuclides or metalloids. The reason is that metals cannot be biodegraded. You cannot degrade cadmium or chromium or lead or arsenic or uranium. What microbe can do? Microbe can to some extent transform it. And many a times we will learn about these things, that microbes many a times they enzymatically they carry out the chemical transformation of the toxic heavy metal compounds. So, basically the metals and radionuclides cannot be biodegraded. So, you cannot consider or compare the degradation of organic pollutants with heavy metal bioremediation process, because metals and radionuclides cannot be degraded. However, microorganisms can interact with these contaminants like the heavy metal radionuclides contaminants, and they can facilitate an array of processes, and these array of processes include redox transformation, that is the change in the oxidation state of the pollutant molecule, maybe arsenic or chromium or uranium or any other metals or metalloids or radionuclides whose the oxidation states are of important factor in regulating their environmental toxicity, environmental significance, their ability to bind to different mineral phases or ability to dissolve and remain in the aqueous phase, for example, complexation, precipitation, absorption and accumulation are the other processes which are responsible for controlling the behavior of the toxicity, the mobility of metals and radionuclides in environment, and these are carried out by the microorganisms present in the environment. So, these are the reactions or the possible processes which are available with the microorganisms to deal with the toxic the heavy metals or similar metallic compounds. So, since heavy metal are not subjected to biodegradation per se as compared to the organic pollutants. So, the remedial goals when we have a contamination by chromium or uranium or arsenic or something like that, these are heavy metals. So, we cannot biodegrade or cannot oxidize them straight away, so that they become decomposed into carbon dioxide or water. However, the remedial goals can still be achieved using microorganisms which will facilitate the precipitation and thus immobilization of the inorganic contaminants like the heavy metals or the radionuclides. The precipitation means they will no more be available in the aqueous phase and they will remain immobilized on the solid surfaces. So, just imagine the groundwater where or the aquifers where the pollutant molecules like chromium or uranium might be there due to some anthropogenic activities, and by some means if you are able to precipitate these uranium for example, or convert the arsenic, transform the arsenic in a form where the modified form of arsenic precipitates or the uranium precipitates and gets immobilized on the solid surfaces like the minerals, then the water which is flowing will remain free of those contaminated or contaminants. The second option could be concentration and thus reduction in the volume of the contaminated matrices. Sometimes, particularly with respect to the toxic radioactive metals and radionuclides, the volume of the waste becomes a very serious concern in the front of the environmental technologist and environmental biotechnologist. So, what we have learned that the volume of the waste can be significantly reduced if we are able to use certain specific microorganisms who can actually bind to these radioactive or the otherwise toxic metals and sequester them, accumulate them or absorb them onto their cell surface, and thereby we can store that bioaccumulated or biosorbed materials in a much smaller volume compared to the total volume of the waste. So, from the waste management point of view, that gives us ample opportunity to deal with the large volume of waste, particularly where the radioactivity or the hazard of the waste are of major concern, because storing few lakhs of gallons or few lakhs of liters of water or contaminated waste liquid is a very difficult task rather than few hundreds of liters. So, few hundreds of liters may be stored or can be managed much more efficiently compared to the few maybe million liters of the waste volume. The third one is the compartmentalization of the metals to a part of the environment in which their harm is reduced. That is another aspect where in the natural, in the in situ condition, we can actually deploy certain specific microbial processes which carry out transformation reactions, and those transformation reactions are enabling the metals to compartmentalize in certain part of the environment, and thereby their environmental harm, environmental mobility or bioavailability is significantly reduced. Now, in this section we are going to learn something about the heavy metals, particularly in terms of the problems which are posed by these heavy metals when the microbes are taken into consideration in particular. Now, most of the heavy metals exist naturally. So, heavy metals are part of the earth crust, and from the early evolution we see that the local microorganisms, local biota, all kind of animals and plants are habituated, they are equipped to handle with the heavy metals, because and most of the heavy metals and many of the heavy metals are required as a trace nutrients like zinc, copper, cobalt, etc., but at low concentrations. These are low concentration, means that the parts per million are below that. So, at very low concentrations, biological systems are equipped to utilize the metals as a part of their essential nutrients, trace elements that we consider as our micronutrients that we consider, but they may pose toxicity or they may cause toxicity if the concentration threshold of the same metals like the copper, zinc, nickel, etc., which are otherwise very useful as a trace nutrients, are present in excess concentration. Now, how the excess concentration appears? Because naturally the earth is not made up of that excess concentration unless and until we are dealing with some ores of mineral deposits. It is basically the anthropogenic activities, particularly the disposal of waste from metal excavation, metal processing and other industrial activities, that actually have led to a huge level of rise in the level of this heavy metal concentration, and which we call a dangerous level, because the concentration of heavy metals are now more than what the biota or the microbes or the plants are naturally equipped to handle with. And in some of these soils and sediments, which are we call them contaminated soils and sediments, and these higher level of heavy metals, toxic radionuclides, metalloids, etc., they are endangering both the wildlife and the people and motivating the efforts to detoxify the soils. So, we are trying to look into the aspects that how natural microorganisms, even plants and fungi, etc., how they behave when they are exposed to the higher concentration of these toxic metals, and how we can learn from them and then develop technologies which could possibly help us to have bioremediation processes. Now, microorganisms, as I mentioned, have coexisted with metals since the early days of their evolution. So, from the day 1 of their journey in this planet, they are living with the metals, because metals are the integral part of the earth crust, and a wide range of divalent and transition metals are present, are involved in the enzymes active sites. So, many enzymes we see, they contain these trace metals like zinc, nickel, cobalt, iron, etc., which are required as a traces condition. And many a times when we grow the cells in the laboratory, we may not add these heavy metals as such as nutrients, because they are supposed to be coming from the other materials that we supply as a growth medium. But in a natural condition, in a natural environment, microorganisms, they just acquire these divalent and transition metals, which are required as a trace ions for them. Now, metals, many metals, as I mentioned, the zinc, cobalt, cadmium, not cadmium, zinc, cobalt, copper, etc., iron, magnesium, for example, could be biologically very useful, because they are the part of the integral part of the enzymes which require many metalloids. For example, we call these enzymes, they require the metals for their activity, but they may be toxic, they may be creating a lot of stress to the organisms if the concentrations are beyond the desirable range, even for the required essential ions. For example, copper or zinc, copper may be trace element required by the cell, zinc, nickel are required by the cells for different enzymes, but if the concentration of the copper or zinc or nickel is elevated beyond certain threshold limits, then possibly the microorganisms and all other organisms will experience toxicity, toxic effect because of this higher concentration. So, at that point of time, their biological relevance will be different, because then they are stressor, they are toxic compounds. Now, some metals that is like we are referring to these metals which are considered to be the trace elements, and they are the part of the catalytic structure of the proteins or other important biomolecules. These metals are therefore required in minute amount for the normal cell metabolisms, and as a result the intake of these metals are highly controlled within the cell. So, as we have learned over the years that within a cell which is naturally growing in an environment, if we have for example zinc, so these cells would like to transport zinc inside the cell. So, how this will be done? For that there will be some transporter. So, this is called metal transporter. So, these metal transporters are of different types. These metal transporters can be of general type. General type means they are capable of transporting any divalent cation of comparable ionic radius and comparable chemical behavior, or they may be highly specific type. Now, these transporters are produced by the cell. So, the cell has the DNA, and from the DNA they encode the proteins, they encode the mRNA, and from the mRNA following translation the proteins are produced, and the proteins are these transport proteins. So, basically the transport proteins are part of the genes which are present in the organism itself. So, these transport essentially, that means is under the control of the cellular process. So, cell essentially can control this. How? Because these are manifested, these are carried out, the transport is carried out by this carrier complex or the transporters. So, these transporters, which may be general transporter or may be divalent transporters or may be very specific transporter for a particular ion like zinc or nickel or cobalt or copper, there may be specific types of. So, in either of the cases the cell can control the expression and the production and the formation of these transporter molecules, and hence they can actually control the transport of the essential trace elements, and therefore, that is considered as that the intake is subjected to intricate homeostatic mechanism. It depends on how much is the requirement by the cell. Like for example, the cell is requiring alkaline phosphatase, which is a zinc containing enzyme. So, if a cell for instance is growing in an environment where a lot of organic phosphate is there. So, if the cell is growing on an environment where lot of organic P is there, and the organic P needs to be metabolized, so that the P phosphate is available to the cell, so that cell can transport the P. Now, for this process the cell will require the alkaline phosphatase and other phosphatases. Now, the alkaline phosphatase particular is a periplasmic enzyme. This alkaline phosphatase will require zinc. Divalent zinc ion will be required for the alkaline phosphatase. Now, if this particular cell is growing under the organic phosphate, in presence of organic phosphate, then possibly a requirement of the zinc will be slightly higher compared to the environment where the cell is not growing under organic phosphate, rather the cell is growing with inorganic phosphate. That means, the cellular requirements determine how much will be the transport of the zinc into the cell. So, the behavior of the cell with respect to the divalent cation zinc is entirely in the hands of the cell itself, that what the cell wants. So, that the sufficient quantity is supplied, but the supply must not be excessive. So, under this condition the cell will maintain the homeostasis, that means the equilibrium, so that the level of the zinc which is accumulated inside the cell will be always linked to the requirements. How much the cell requires, that will be the major criterion for determining how long this transportation will occur. So, under certain other environmental conditions, some other heavy metals, we will come back to the zinc maybe very soon, but maybe some other metals, they seem to have no biological relevant function, for example, cadmium. So, for example, cadmium if there along with zinc, then cadmium is not required, because cadmium to CD2 plus ions are not required by the cells. But by some means, if the cadmium 2 plus ion is capable of entering in the cell, then that cadmium is undesirable for the cell, and that is going to cause harm or damage to the cell. Why? Because these heavy metals, either they are non-essential, they are not required at all. So, they are fundamentally identified as non-essential. So, they are not required for the cell, or they are required, but at much lower concentration. So, now you have two situations. In one situation, so in one of the situations you have only the ion which is required by you, like zinc 2 plus. You need zinc 2 plus, so you have a transporter and the zinc concentration is controlled, but that zinc should be at the nanomolar or micromolar level. So, maybe it is close to or around some nanomolar level, around 1 nanomolar or so, that is sufficient for the cell. But in the environment you might have a millimolar level or maybe 5 to 10 millimolar level zinc. So, in that case the concentration is so high compared to what is required. In this condition the zinc is toxic, but in this condition zinc is non-toxic. So, the cellular response or cellular homeostasis will be adjusted in such a way that when the concentration threshold of the desired heavy metal is met, that means the concentration threshold is below the toxic level, the cell will deploy a type of response to the heavy metal, because the zinc is already identified as an essential, but and its level is also in the non-toxic level. So, that time the cellular response will be different, but when you have much elevated zinc, no matter whether the zinc is trace element required by this bacteria or not, only because the concentration is so high, then the zinc become toxic, and then the cellular response will be something different, that we will study, that what happens when the cells are exposed to a particular element which is already a trace element required by the cell, but in a given environment it is present in a very high concentration. That means, at this point of time the concentration of this particular element or the heavy metal is of a point of concern. Now, there is another situation where you have cadmium which has no biological function. So, what will happen? So, cadmium will obviously be not desired inside the cell, cell has no desire to take cadmium inside the cell because it has no biological role, but the cadmium might try to enter inside the cell. This is the property of the cadmium because it might resemble the transporters which are general type divalent cation transporters, and those divalent cation transporters can be utilized by cadmium to enter inside the cell, and then the cell will respond differently. So, cellular response will be different, cell will not be happy at all because the cadmium at any level it is toxic. Now, cell will have its own mechanisms to combat this situation. If the concentration of the cadmium is low, like in a nanomolar level, then the response will be something. If it is millimolar level, then the response will be different, that we will see later. So, now from the physiological point of view, we are able to then categorize three types of the metals. One is essential like zinc, cobalt, all these things will come later, but some are even more general type like calcium, magnesium, and these are called essential, but basically non-toxic. So, even if the concentration goes slight higher, the cell will not mind because these are not toxic, they do not have the similar type of effect that otherwise the heavy metals like zinc, copper, cobalt might have. The second category is the essential, but harmful at higher concentration like iron, manganese, zinc, copper, cobalt, nickel and molybdenum. They are required, but at low, very low concentration. If the concentration level crosses that threshold, which varies of course from cell to cell, different environment also, they become toxic. And the third one is obviously toxic, they are always toxic like mercury and cadmium. Now, microorganisms have evolved a whole range of mechanisms of interactions with these metals to ensure the adaptation of microorganism to a changing and frequently hostile environment, because the environment is not static. So, environment is changing always depending upon different physicochemical conditions, the level of heavy metals are also going to be changed. So, microorganisms are equipped to deal with such situations. So, for example, a bacterial cell is shown here, and you can see one M is a metal ion. So, in one case the metal may be required by the cell. So, cell would take up this metal and utilize it. In other case, the cell will not take up the metal and will just discard this, cell will not take it into inside the cell. So, that means the same cell has different mechanisms of interacting with the metal ions. Now, the emergence of the metal tolerance determinants, that is the genetic determinants, seem to have been very early event in the evolution. Ever since the microbial cells are experiencing the different concentration thresholds or concentration levels of different heavy metals, they are learning that how to deal with that. So, they have initially adopted maybe some general strategies, and then they have started evolving some specific methods of dealing with the heavy metals that we see today. And in more recent time, particularly with the industrial revolution and all those things, the anthropogenic mobilization from the metals of metals. So, different metals from diverse waste materials has created novel metal loaded niches with strong selective pressure for metal endurance. So, therefore, in such niche where we have more heavy metal concentration, we can expect that the microorganisms living over there, they might be deploying more intelligent and more efficient mechanisms. Now, if we look at the overall spectrum of the different microbial or bacterial abilities to deal with the heavy metal different type of heavy metals. So, this is a kind of a general plan that with a concentration varying from nanomolar to the molar level, the organisms like eukaryotes or cyanobacteria, they have a very specific type of way of dealing with this, like using the metallothioneins or some kind of these proteins are utilized to deal with the toxic concentration of the heavy metals, which are of course at very low level. At moderately higher level, the bacteria, particularly talking about more on bacteria, the gram positive and gram negative different bacteria, including the E. coli and Desalpo-vibrio and many other members, they deploy a large number of systems, a variety of systems, which include the efflux systems that we will learn, the enzymatic transformations which are very specific to specific toxic metals, and metalloids, and at very high concentration of metals which are at molar level, which are more frequently encountered in the mine sites, mostly the metal mine sites. We see that very high level of metal concentrations are prevailing. So, acidophilic microbes and chemolithotrophic bacteria, they deploy again another specific set of mechanisms which help them to deal with this very high concentration of the toxic metals, and of course, that include the precipitation as one of the major means. Now, several prokaryotic organisms, bacteria and archaea, they show specific resistant determinants. So, these processes are often very specific. So, as I mentioned earlier, some mechanisms might be general, like production of metallothionein or similar in plant system we see the phytochelatins, something comparable to that. But this kind of sulfur hydryl group containing small protein molecules have been found to be very efficient at low concentration thresholds, but at higher concentrations the microbes, the bacteria, they deploy a large number of systems. So, and the specific genetic determinants for that type of resistance systems or transformation systems are all very well studied. Now, microbial studies, microbial activities that affect the metal speciation and transport will be discussed. The different organisms exhibit diverse response to toxic ions, which confer upon them a certain range of metal tolerance. So, they know how to deal with this toxic metal concentration. So, one idea that I presented is the metallothioneins, which are basically a family of metal chelating proteins, are mostly used by eukaryotic organisms, and cyanobacteria we have found that they are also capable of producing these metallothioneins. So, it is mostly an eukaryotic mechanism, mostly eukaryotic mechanisms, but in some prokaryotes they are also present. But they are more sensitive to metal toxicity, particularly towards the lower concentration regime. And what we have seen that eukaryotic organisms are more sensitive than the prokaryotes. So, prokaryotes they use a whole set of different mechanisms compared to the eukaryotic sets, which are capable of producing these metallothioneins for combating the low concentration of heavy metals, and that actually help the cells to regulate the intracellular metal, because these contain the sulfur hydryl or SH groups. These SH groups are capable of binding the divalent metal cations. So, forming the metal sulfur bonds, and the metal sulfur bonds are used to chelate to precipitate the metal inside the cell. So, that the metal divalent metal ions are not available for interaction with any other cellular components. Now, MTs as or metallothioneins as the main mechanism of metal tolerance is exceptional in the bacterial world. Generally, we do not see much of bacterial interest in producing this metallothionein. So, what bacteria do? Bacteria they deploy a number of more specific, because metallothionein is not a very specific mechanism, that is more towards a broad divalent cation or divalent heavy metal homeostasis mechanism. Bacteria they deploy active efflux, sequestration and redox mediated different transformation processes, through which they try to withstand the toxicity, and also often they detoxify actually the metals. Now, these mechanisms are functional at concentration above the homeostatic or non-toxic level. So, like for zinc or copper or nickel, which are actually required essential trace elements, for them this kind of like efflux pump may be operating when the concentration goes beyond certain threshold limit. Or for cadmium even or mercury, we can see that the transformation or the active efflux is actually operating even at much lower concentration. So, it depends upon what are the metals, what are the type of microorganisms, and in which environment they are actually interacting. Now, in context to bioremediation, this metal microbe interaction is developed as a part of interdisciplinary research area, where we see the assemblage of microbiology, along with that geology, geochemistry and molecular biology, because essentially these microbes they need to interact with these metals in a practical context, which will have lot of geological parameters, geological factors within that. And we have a number of benefits from these, and these could be actually the micro to nano scale characterization of the microbial communities in their natural habitats, to understand what type of microbes are involved with different metals, what are their responses, how actually they are dealing with the toxic metals and radionuclides. On the other hand, the characterization of the cell associated metal complexes, because often the metal ions are subjected to change in their oxidation states or change in their other chemical properties, like including their this solubility in the aqueous medium. So, under such conditions, how and what are the properties of those deposited metals or sequestered metals and their stability, etc., those are also characterized. And an understanding of how microbes affect the partitioning of the metals between the gaseous, solid and the liquid phases in situ, because in a practically the heavy metal pollutions will be in the outside world. How exactly microbes will be capable of partitioning the metal, so that they are not coming to in the aqueous phase, and that is emerging to provide a scientific basis for the metal and radionuclide bioremediation. So, in the general way, if we look at this, so microbes, here the microbial cells are one of the cells I have drawn, and the M2 plus is indicating the divalent metal ions. So, as you can see, there could be 4 basic mechanisms that I try to put here. So, these 4 mechanisms will begin with, first I broadly categorized into bioaccumulation and biosorption, although this could be again splitted into 2 events like bioaccumulation and biosorption. But the idea here is that in case of bioaccumulation, the divalent cation is transported inside the cell, either using some general transporter or some specific transporter. By some means, the toxic metal ion will come inside the cell and will begin accumulation. So, you will find that large number of divalent or the metal ions are deposited or accumulated within the cell. So, that is called the bioaccumulation process, that is a part of the interaction. And of course, this is a property of a living cell, because lot of transporters will be engaged, and these transporters sometimes they need energy also. So, we need to spend ATP for that or other transmembrane gradient. So, we need to spend energy in some other form, either ATP or the transmembrane gradient. The other one is biosorption, which is more like a physicochemical process, where whether the cells are living or cells are non-living is not that important. Biosorption is a physicochemical process where the divalent cations will be physically or chemically or both physicochemically, they will be sorbed. Now, the word sorbed means both adsorption and absorption are included in that, and in this case the bacterial or the cellular structure, the cell envelope plays a very important role, because that is full of different negatively charged groups, and those negatively charged groups they allow the divalent cations to sorbed. So, that includes both the adsorption and then absorption. So, adsorption is on the surface and then gradually it moves in. So, now if we imagine that is the outer membrane and you have the periplasmic space or the cell wall part. So, you have the cell wall and a gram negative bacteria, you have also the inner membrane. So, the heavy metals will gradually enter and reach up to the inner membrane. Sometimes they have been found that they are completely loaded, the cell envelope region which is covering entire piece of this outer membrane, inner membrane and the cell wall part, that is called the cell envelope, is totally loaded with these metals, because it is both the adsorption first and then absorption. This is a physicochemical process. We will discuss about this later. The second and very important and interesting phase is the biotransformation, which is found to be mostly redox driven process. Oxidized and reduced, either oxidized or reduced species are converted to some other form. For example, if we take an example that arsenic 5 plus for example can be reduced to produce arsenic 3 plus, and vice versa. So, arsenic 3 can be oxidized to produce arsenic 5. Now, when the microorganisms change the oxidation state, or uranium for example, uranium one form to the other. So, when we do this kind of what we see, the microorganisms are doing this kind of redox mediated transformations, or maybe for chromium also, or for iron or other redox active metals. So, that leads to some kind of change in the chemical behavior, because if we compare arsenic 5 and arsenic 3 is more toxic and more mobile compared to arsenic 5. So, arsenic 5 is relatively stable compared to that. Now, possibly then the microbes will not try to do this reaction, because if they are doing this, then possibly they are generating more toxic species. But then there could be some other bacteria who can again transfer this arsenic 5 into arsenic 3 into arsenic 5 through oxidative reactions. So, you can have both reductive reactions and oxidation reactions. So, all kind of redox reactions are possible in the environment, depending upon the redox conditions and the type of microorganisms. So, the idea is these the biotransformation type of reactions, they play a very important, for example, the chromium, for uranium, we have seen that lot of successful bioremediation process have been developed. The third one is the biodegradation. It is not the degradation that we have learned during organic pollutants, but this is rather the degradation where organometallic complexes, because metal organic complexes are found to be more toxic, more mobile, they move very fast within the environment, they are accumulated in the living cells very fast. So, often microorganisms are capable of breaking down this organometallic bond. For example, methyl mercury. So, if you break the methyl mercury, that bond between the mercury group and the methyl group, then the mercury becomes less mobile compared to the methyl mercury molecule, and then similarly for the arsenic also. So, microorganisms often they deploy some enzymes to break down the bond and then deal with the metallic element separately. The fourth one is the extracellular complexation or precipitation by different cellular ligands or cellular molecules produced by the microbes. For example, polysaccharides, metallocyanins or siderophore like compounds, which are produced by the cells and they are released. So, once they are released, these molecules, when they are released or produced outside the cells, they are capable of extracellular complexation and precipitation of the metals. So, now if the metals are accumulated in the cells, in the case of bioaccumulation that we have learned. So, what happens to the accumulated metal? For that also we see that there are a number of strategies developed by the microbial cells, the bacteria in particular. So, one of the most prominent ones is the efflux, where the accumulated metal is thrown out or exported outside the cell by spending energy like either ATP or proton motive force or other source of energy. So, that the cell is free from the metal. In other instance, we see that cell is capable of compartmentalizing the metal ions. For example, in case of copper, we have seen some bacterial strains of Pseudomonas, they are able to sequester the copper only in the periplasmic or the cell envelope space. So, that is a kind of their strategy, they produce a specific type of copper binding proteins only in the cell envelope region, and this cell envelope region copper binding protein they bind the copper and then allow the compartmentalization of the copper only in the periplasmic region. So, that the cell interior is free from the copper, otherwise the copper will have its toxic effect and the cell may not be feeling good with that the stress. So, this is kind of all the strategies that the cells are developed or they execute. And the other one is the enzymatic conversion. For example, with chromium, with arsenic, we see that there are different type of enzyme, mostly the reductases type of enzymes, which converts this metal, toxic metal like chromium for example, to a form which can be effluxed out. So, in that case there may be like mercury can be reduced, and then the mercury can be reduced Hg0, Hg2 plus to Hg0, and Hg0 can diffuse out of the cell, or can be a efflux pump can be there, that efflux pump will help the help the cells to actually transport out. For example, in case of arsenic, we see that there are cytosolic arsenic reductases which actually reduces arsenic 5 to arsenic 3, and then arsenic 3 is effluxed out by a specific transporter pump. Alternatively, the cells can also produce certain small protein molecules or a few amino acid chain molecules to sequester the metals inside the cell. Maybe the metallothionein is one of these examples. So, again the metal is sequestered and unable to interact with anything else inside the cell. In case of bioabsorption, which we have already discussed, that is a metabolism independent physicochemical process where a large number of functional groups present within the cell envelope regions, including the carboxyl, sulfonate, phosphate, hydroxyl, etc. they are involved, and it involves basically a large number of processes, but among them the ion exchange and adsorption microprecipitation have been found to be most prominent in case of the bioabsorption process. Just remember that this is including both the absorption and adsorption, both type of processes are there, and it is metabolism independent process where the cells need not be living when this bioabsorption. So, from a bioremediation point of view, there have been a considerable research interest and process development have been done because they can be performed using dead or metabolically inactivated cell also. Now, in case of the microbial mediated redox transformation, that is called biotransformation reactions, the oxidized forms of different metals or radionuclides like uranium, technetium, chromium, etc., can be reduced to some form like uranium 6 to uranium 4, chromium 6 to chromium 3 or technetium 7 to technetium 0, and all these will lead to altered mobility. So, they are actually prone to precipitate. They are prone to precipitate in the aqueous environment where the pH is around neutrality. So, you will find that almost the aqueous phase is free from toxic uranium or toxic chromium, because most of the uranium and chromium they have been precipitated in the solid phase. So, the water which is flowing will be free from these ions. Another aspect is the reduction of the sulfate. Sulfate reduction leads to the production of sulfides, and these sulfides can react with divalent metal ions and further produce the metal sulfides, and metal sulfides they precipitate. So, that is another means by which microorganisms they interact with these toxic metals and radionuclides. Alternatively, these metals and radionuclides can be oxidized also. So, a large number of metals, uranium including uranium 4 or cadmium sulfide, zinc sulfide, arsenic, etc., we see that the reduced species are subjected to oxidation, and then the oxidized forms are produced. But we need to remember that again the oxidized forms might have different environmental behavior, but whether oxidation will proceed or reduction will proceed, that depends on the environmental condition, particularly the redox condition of the environment itself, and also the microorganisms which are present over there. So, for this part of the lecture, the following references can be used, and in conclusion we have discussed the basic concept of metal microbe interaction. Mechanisms of microbial interaction and their relevance to bioremediation is highlighted, and the concepts of bioaccumulation, bioabsorption and biotransformations are also discussed in today's class. Thank you so much.